Yeah, and now we get started, as you heard. So have fun with the lecture of Anna, and thank you once again, Anna, for making yourself available and give us this lecture.
Thank you so much, Tatiana. I'm really excited to be here. I'm just going to do the requisite sharing of the screen before getting started. All right, so then we can also get a laser pointer moving there. So thank you so much. It's just so wonderful to be here over Zoom and share with you some of the science that we've been doing at the University of Chicago over the past few years. But before I get started, I really just want to take a moment to thank the Bayer Foundation for everything. It's been so wonderful to meet the community in Leverkusen in February in person and over many exchanges online. Thank you so much for meeting with me and sharing amazing work that the foundation is doing and also Bayer in general. I'm learning so much, and it's just it's such an honor to be part of this community and be recognized with this honor. And my group and I are very excited for the chance to share with you some of the work that we've been doing, and hopefully you'll find it interesting. So what my group has been working on over the past few years is the development of reactions that are driven by electricity across what we call the chemical value chain. So taking low value added reagents and then making them into more complex value added products. The cool thing is that if we do this and we drive these reactions using electrons, these electrons can ultimately come from renewable sources of electricity, like solar and wind. But of course, the sun is not always shining and the wind is not always blowing, so these sources are intermittent. But if we use this intermittent source of electricity to drive these chemical reactions, you can contribute to two primary scientific goals. The first is solar fuels. So you can store this energy in energy-dense chemical bonds, so you can retrieve it when you need it later, such as engaging in what we would call the reverse water gas shift reaction, taking CO2 to make carbon monoxide, which is a fundamental feedstock for Fischer-Tropsch chemistry. Another main place where we can contribute is sustainable synthesis goals, very much at the heart of what Bayer is actually working on. And here I've shown two Bayer molecules where electrosynthesis methods have been established also. Waldvogel and co-workers in collaboration with Bayer have shown us that you can, instead of using toxic oxidants, you can use electrochemistry, just electrons and holes, to produce this herbicide safe thinner in one simple step. Additionally, recent work from Bayer has shown us and taught us that you can do recycling of the undesired enantiomer of this receptor agonist using electrochemical reduction, further showing us the power of electrosynthetic methods in enabling the process of manufacturing agrochemicals and pharmaceuticals. But what I want to stress is, despite the diversity of all these types of reactions that one could do using electricity, they're all unified in two primary ways. The first way that they're unified is that all of these reactions occur at a phase boundary between a conductive solid and a liquid electrolyte. And it's at that phase boundary where all these electrochemical reactions occur. The second thing that unifies all of these reactions, despite the diversity of the transformation, is that they're all fundamentally limited by what we would call Faraday's constant, 96485 coulombs per mole electrons. And that limits us because if we wanted to electrosynthesize, for example, a mole of product P, and we had a typical lab scale working electrode that can conduct the electricity about 5 centimeters squared in size, then it would take us 44 days, a very, very long time to make that amount of product. That's a really long time. And so there are two primary knobs that we can tune because of this fundamental equation of current, time, and area that we can use to increase the productivity of your desired chemical transformation. The first knob you can tune is, you can imagine, to use the biggest surface area working electrode you could ever imagine. And the second knob that you can tune is to augment the current. And augmenting the current is a cool way to think about it because it's a chemical means that you can actually tune. Why? This is because the current, which is the flow of electrons, which I have denoted as J sub P, is directly proportional to the velocity of the reaction. And we know through a fundamental relationship that we learn in general chemistry, the Eyring equation, that there's an exponential relationship between the activation energy barrier and the rate of the reaction. So as I increase the driving force of the reaction, or the applied potential to my electrode, I decrease the activation energy barrier for my reaction, going from substrate to product, and involving electron transfer. And so what you get is a exponential dependence between the applied potential and the observed current, i.e. the reaction rate. But at some point, you'll be going so fast that now you're limited by the diffusion of the substrate to the electrode surface. So what you end up getting is an activation controlled potential regime and a mass transport controlled potential regime. So remember, what we want to do is augment this current density because we want to increase the productivity of our electrochemical synthetic reaction to make agrochemicals and pharmaceuticals. So what are some ways where we can simply just augment that current density? The first is the one way to do this is to just use catalysis. Catalysts are things that make reactions go faster. You can imagine that you had a homogeneous catalyst in solution, and if you had a homogeneous catalyst in solution, it would increase the reaction rates. But you're still fundamentally limited by the diffusion of that homogeneous catalyst to the electrode surface, so you can engage in the actual catalysis. So another way you can make these reactions go even faster is if you fix the catalyst onto the surface of the electrode. So now the electrode material itself is the catalyst, and now you can further augment reaction rates this way. And what's cool is that now that the surface itself is the catalyst, the conductive surface, then you can imagine this is a reusable material, and you can use it for several different reactions. And it could, in principle, be readily adopted by different types of chemists. So you might ask yourself, why aren't we using many different types of heterogeneous electrodes for different types of electrosynthetic transformations yet? I think this might be one of the reasons why. When we typically think about catalyst design, we typically think about modifying singular, uniform active site. Every single catalyst in my flask is the same as its neighbors. And because of this uniformity, what I can do is imagine a molecularly controlled, precise modification before I even go to the lab and make it. And because I can imagine it, I can use the power of molecular synthesis to actually realize that proposed modification. And then I can test my catalytic hypotheses to inform my next design, and then iteratively optimize the structure of my catalyst to achieve my desired function. But now, if we want to use heterogeneous catalysts to increase the productivity of the reaction and also make it more reusable and integratable into devices, now we're looking at catalysts that look more like this. Your catalyst is now dispersed on the surface of the electrode. So I don't even know where the active site of my catalysis is. And in a lot of the state-of-the-art electrocatalytic materials that have been reported, particularly for small molecule activation, many researchers have shown that these surface structures are incredibly non-uniform. So every single active site is not the same as its neighbors. It's incredibly disordered. So how can we similarly imagine systematically controlled modifications of these complex, messy systems? Wouldn't it be great if we could similarly have this type of predictive molecular control so we can systematically and iteratively optimize these types of complex, messy systems to increase our desired function? And I think this is an incredible challenge in this field, and it's one of the primary motivations of my research group. So at the University of Chicago since 2021, my group has been working on thinking about this problem a lot. And we've been doing several different experiments. The first is we're developing material synthesis strategies that are compatible with in situ spectroscopies, so we can actually characterize the heterogeneity or the disorder of how molecules interact with surfaces. And then we're using the power of molecular synthesis because we love molecular synthesis, but now we're combining it with non-covalent self-assembly. So maybe we don't need to know where the active site is or even what it is. And I'll show you an example of this today. And then finally, we're using the power of mechanism to guide our design and actually achieve selective electrosynthesis across a wide array of different reactions. And I'll show you two examples today. So let me first show you what my group has been doing in terms of this fundamental problem of how do we design for disorder at the surface of these electrode materials. We're really inspired by nature. So nature is an amazing, is amazingly creative. So if you wanted to do a reaction in physiological conditions, so that means that you're at neutral pH conditions. If you had a residue like glutamic acid and its pKa was around 4, then these carboxylic acid groups would be fully deprotonated. And you can't really engage in any sort of meaningful acid catalysis at the neutral pH environment. What's really cool is what nature has done is that has evolved active site environments that can make tailored microenvironments around these residues. And in doing so, you can shift what is the fundamental value of pKa by around 2 units, for example. So now you can do acid catalyzed reactions even under the constraint of physiological conditions. Imagine now you wanted to do electrosynthesis in water. Water is, of course, neutral most of the time. And also, it's one of arguably the most accessible solvent for synthesis. So we wanted to test this idea. And we wanted to test this idea with some model reactions so we can understand the design principle or how to get to the design principle. We chose the electrosynthesis of hydrogen peroxide, which is a key oxidant that's used in a variety of different types of reactions. In fact, this technology that I'll tell you about today was established in Germany. It's called the anthraquinone process or the Rieder-Federers process. You take anthraquinone and you hydrogenate it to form hydroanthraquinone, and this can react with molecular oxygen to make hydrogen peroxide. You could imagine that hydrogen, which consists of 2 hydrogen molecules, so each of them has an electron and a proton, you can simply just replace hydrogen with 2 equivalents of protons and 2 equivalents of electrons, and these electrons can come from electricity. And then you can engage in anthraquinone-mediated or catalyzed O2 reduction to form hydrogen peroxide, and many people have done this. But it's interesting to note that there's not many reports out there that works in neutral media. It's kind of like this enzymatic analogy I showed here. Wouldn't it be great if we could establish design criteria so that we can similarly have this chemistry work even in neutral media? And then it would unlock some sort of way to design for this disorder. So one of the reasons perhaps why this is not as reported in neutral media is because the pKa of hydroanthraquinone, which is proposed to be the active form, lies at around a neutral pH value. So you can imagine you have a significant population of the deprotonated form. But if you had some sort of synthetic technology that would allow you to tune the pKa of this hydroanthraquinone, like in an enzymatic system, then maybe you could engage in this chemistry even under neutral constraints. So let's see if there's something that we can do here. So of course, there's many ways to attach molecules onto electrode surfaces. And one of the most powerful ways of doing this is covalent modification. Of course, this is not an exhaustive list of the type of technologies that are out there, just representative. But what you can do is you can, for example, if you had a thiol or carbene that is attached to a molecule, this as denoted by this orange square. So this orange square could be your anthraquinone catalyst. You can attach it to the surface of a conductive material or an electrode. And this synthetic technology is super cool because it's predictive in its nature. You can go to the lab, you can imagine the modification and go to the lab and make it and expect it to attach. But this type of covalent technology is challenged in two primary ways. The first is that it's highly specific to the surface chemistry of the electrode, that material that you're using. Things like thiols work with gold, carbenes work with palladium, so on. And while there are diversity of different types of materials that work, they don't work for many, many different types of electrode materials, especially messy, disordered materials. And then another thing that's challenging about these covalent modifications is that they're designed to be permanent. There is not really a mechanism of repair built in. But many researchers have shown that they irreversibly desorb or degrade under electrochemical conditions. So wouldn't it be great if we could have a more general way of similarly molecularly modifying electrode interfaces that is predictive and also has a mechanism of repair built in? So my group has been working on this for the past few years, and we've thought about a strategy that's a little different. What about instead of covalent tethers, we use non-covalent technologies? What if we had in all electrochemical systems, we always have a built-in non-covalent interaction, and that's electrostatics. So imagine you had a negatively charged electrode. If you had a positively charged cation in solution, like this ammonium, you could imagine that it would go to the interface to form the so-called diffuse double layer. But the challenge is, what if you reverse the potential bias from negative to positive? Is electrostatic repulsion going to repel the layer? What we figured out is if you synthetically stitch on this long aliphatic tail, even if you reverse the potential bias from negative to positive, force other van der Waals forces called van der Waals between these amphiphiles can override electrostatic repulsion and keep the layer there. The second idea is that these amphiphile molecules, meaning it has a polar head group and an aliphatic tail, essentially it's like soap and water, form aggregate micelles in bulk aqueous solution. And it's also well documented that you require dynamic self-exchange between the monomeric form and the aggregate micelle form to maintain the thermally equilibrated state in bulk solution. So we wondered, is there also a reversible linkage here that we can hijack to render these layers to be regenerative, even if we intentionally degrade the layer? So what my group has been did was we made this model amphiphile system, and we put on this ferrocene redox active tag so that we can track the layer growth. And our results are consistent with a cartoon model that looks like this, where the polar head group does go to the surface, and then these molecules sort of stack like little trees in a forest. And we are able to use this ferrocene redox active tag to ask ourselves if this layer formation works on different types of electrode materials. In other words, is it general? And we see that this works for different noble metals, palladium, platinum, dilutely modified gold, or even nanoparticulate morphologies. It works on different surface terminations of carbon, glassy carbon, highly oriented pyrolytic graphite, edge plane pyrolytic graphite, boron doped diamond, which are very important classes of materials for electrochemistry. And also different types of functionalized carbon, this disordered carbon that I was talking about. And nanoparticles also on carbon also work. And we are tracking this redox active tag of the ferrocene to establish this. And while I'm glossing over spectroscopic data and other analytical data that we use to corroborate this hypothesis, what I wanted to stress is we find this really cool because now you can imagine you're taking the chemical space that's already accessible with ex situ organic synthesis of the amphiphile. And what you're doing is you're combining it with non-covalent self-assembly, and you can now also access the electrode material space, which is anything that is conductive in principle to be able to mix and match and modify the surface chemistry of your electrode. So the way we build these amphiphile structures is through a multi-step synthesis, but I wanted to stress that it's modular so you can change the dynamics. I started out by saying that what if we could regenerate the layer? Well, what my student Nico found is that you can change the alkyl tail length that you're starting with, this bromide, and in doing so, you can unlock different types of dynamics at the interface and thereby unlock the regenerative function that we hypothesize. So it's molecularly controlled function. Using synthesis, we already know how to do. And now if we go back to the original hypothesis of the hydrogen peroxide synthesis, you can imagine now that we can change this orange square from ferrocene to anthraquinone and ask the question, can we similarly use our non-covalent self-assembly to pin the anthraquinone in this on the electrode surface? So what Greg, Deepak, and Ashok did was exactly this question. We needed some sort of control where the anthraquinone doesn't assemble on the surface, and then of course the non-covalent self-assembled variation. So the control is simply just chop off the long aliphatic tail so that we hypothesize it cannot form these layers at the surface. And of course, here we have the one with the long tail where we have the anthraquinone embedded in. Now, if you look at the redox chemistry in neutral aqueous media, these, what we would call voltammetry features, are consistent with localization of the anthraquinone moiety at the surface and diffusive behavior for the one without the long tail. You can use a technique known as surface-enhanced infrared absorption spectroscopy, which is essentially a technique that you can use to investigate the nanometer distance within 8 nanometers of the electrified interface to ask questions about what the surface actually looks like by tracking the vibrational spectroscopy that's present at the interface. And what you can immediately see is that for the one with the long tail, you see lots of different features, and you see a huge bleach in the regions corresponding to interfacial water, which means that indeed these layers can repel off water like the enzymatic system. And then you can also see that you don't really see any features for the diffusive control consistent with them being not really localized or arranging at the electrode surface. You can also count the number of charges of how many anthraquinones are actually on there and compare it with computational findings and suggesting that you're getting these monolayer coverages of the surface of the anthraquinone at the surface. The simple fact that you can see these redox features suggests that you can do a multi-proton, multi-electron reduction of this anthraquinone moiety to hydroanthraquinone, and that suggests that proton delivery and egress is facile through these layers. What's super cool is that we believe that this is still occurring via non-covalent self-assembly mechanism. So if you have the electrode surface negatively charged and you add in the molecule, you see all the spectroscopic features consistent with a layer formation. But now if you intentionally charge the electrode positively and add the molecule into the bulk, you see a featureless IR spectra, suggesting that you're not making the assembly when the electrode is positively charged. What we can now do is ask the question if we're actually changing the pKa of the residues when we do this non-covalent self-assembly. So what we can do is look at the what we would call the diffusive control first. So we're not going to look at the non-covalent self-assembly. And because of the structural uniformity of these anthraquinone moieties, what we can do is use voltammetry to estimate these values. When we reduce anthraquinone to hydroanthraquinone, it's not a simple process. You can go through multi-electron, multi-proton steps, which we would call proton-coupled electron transfer. And it would form this, what we would call a 9-component Swirsky. And what we can do is use voltammetry, electrochemical methods, to go to the lab or Greg went to the lab, my student, and construct a series of voltammograms at different bulk pH values. And then you can reanalyze these data, take the apparent redox potential of these as a function of the bulk pH, and you see this sort of curved result. And then you can use something called and then you can estimate the slope of these values. And that gives you the stoichiometry of the multi-electron, multi-proton value chemistry. And then you can use electrochemical simulation and theory, electrochemical theory, to estimate the pKa values as well as the redox potentials associated with this 9-component square scheme. And what's cool is now, if you take a slice at the neutral pH values that we're targeting for this model hydrogen peroxide synthesis, the data are consistent with us going through what we would call an ECE scheme. And the pKa of hydroanthraquinone is indeed around neutral pH value, suggesting that we are accessing primarily this deprotonated form. And now we want to do the same experiment, but with a non-covalent self-assembled variation. You can similarly now take the voltammograms as a function of pH, and then do a similar analysis to the one I described earlier, and divide it into different electron and proton transfer regimes, do simulation and analysis to estimate the redox potentials and pKa values. And what's super cool now is that at the targeted neutral pH values for our model hydrogen peroxide synthesis, you can now see that we are primarily now going through an ECEC mechanism because the data are consistent with the pKa of hydroanthraquinone being shifted by 3 pKa units. So that suggests that unlike the diffusive control, you can access the protonated form under neutral pH values. So can we simply just translate these catalyst layers onto electrode surfaces that aren't very good, or are catalysts themselves, and do some hydrogen peroxide synthesis? So all of these data now are only going to be at neutral pH values. So we hypothesize that for the diffusive control, you're getting 2 electron, 1 proton reduction to make the deprotonated hydroanthraquinone. And for the non-covalent self-assembled variation, you're getting 2 electron, 2 proton reduction to get the hydroanthraquinone variation. So here are the voltammograms of both molecules without any oxygen around. This, we would use argon as the control. And then if we bubble in some oxygen, and now we're transferring these layers onto carbon electrodes that don't have that much convoluting catalysis themselves, we do indeed see for the blue trace, which is the non-covalent self-assembled variation, an enhanced current associated with oxygen reduction. And so we wondered, is this due to the fact that you simply have more anthraquinones packed at the surface, or is it consistent with this pKa change, or both? So the experiment that we did was investigate how much this enhancement is affected by adding in more of this diffusive molecule to mimic the increased concentration of the anthraquinone that is pinned now at the electrode surface. So when you do this titration experiment, you do indeed see that now the diffusive one can be at the similar ballpark of current for the oxygen reduction reaction. But you can see that it cannot reproduce the earlier onset in the electrochemical reduction, suggesting that indeed the pinned pKa value in this non-covalent self-assembled variation is key for changing and is responsible for this shift. This is further corroborated by additional control experiments where we looked at simply just looking at the unfunctionalized amphiphile without any anthraquinone and then adding in the anthraquinone. You cannot get the same shift. This really suggests that you need the pinned version to shift the pKa and get the earlier onset of catalysis. So what have we done? We've simply just taken a known catalyst system and attached it to the amphiphile using organic chemistry. That's ex situ. And what's cool is now we've programmed the function of the catalyst and the local microenvironment, the hydrophobicity around the anthraquinone inside that synthesis. And the in situ self-assembly just happens by itself. We don't have to do anything. You just add in micromolar amounts of this into the aqueous electrolyte, and then you get the non-covalent self-assembly to occur. And if we look at the hydrogen peroxide synthesis for the diffusive control, you can get pretty good selectivity for hydrogen peroxide, but not very good productivity. And in fact, you have to really increase the concentration of the molecule in the bulk solution beyond the solubility limit of oxygen under these conditions to get any really viable current densities. But now if you do it with non-covalent self-assembled variation, you can maintain the good selectivity, but increase the productivity. So you can really, really decrease the catalyst loading and increase the productivity of hydrogen peroxide electrosynthesis under neutral media. We also wanted to demonstrate in this model system if we can recover the catalysts so that we can continuously reuse them somehow. That's one of the hallmarks of heterogeneous catalysis, right? Is this a molecular system that where we lose the catalyst, or can we somehow establish a recovery mechanism? So we wondered if we had an electrosynthesis like this, where we have oxygen coming in, hydrogen peroxide coming out, and we can use a spectroscopy technique known as UV visible spectroscopy to monitor the electronic transitions of the molecule of the catalyst itself and also of the product. You can convince yourself that you have hydrogen peroxide in the solution, even though there's overlapping peaks here, by using something called a copper DMP assay, and it gives rise to this very noticeable UV vis absorption peak here as well. Now, how can we separate out the catalyst? So if you simply just filter it through a cell light bed, we see spectroscopic results consistent with hydrogen peroxide being separated and no catalyst being pushed through. And you can see the hydrogen peroxide also is remaining even in the copper DMP assay. And this allows us to quantify that we can separate around 87% of the synthesized hydrogen peroxide. What about, importantly, if you use the diffusive control without the long aliphatic tail? These molecules are inseparable. You cannot separate them. But then we wondered, can we then recover the catalyst and use it again? And the answer is yes. So you can then take an alcohol and wash through the solite bed, and you can recover the spectroscopic features associated with the pi to pi star transitions of the anthraquinone moiety, and you're not dragging the hydrogen peroxide through with it. You see it featureless in the copper DMP assay. And then you can use these data to quantify how much of the anthraquinone you can recover, and we can recover around 97%. So that suggests that maybe we can fill in this gap with something that looks like this. We can use non-covalent self-assembly as a strategy to molecularly define electrochemical interfaces so that we can start to now think about more predictive design rules to establish electrosynthesis strategies under these benign neutral conditions. Of course, hydrogen peroxide is only a model system, but I think it is demonstrative of what we could do further in the future. So I hope that I've shown you why I'm really excited, or my group is super excited about these non-covalent self-assembled layers. We believe that they can be general and modify wide classes of different electrode materials, that they're not designed to be permanent, so they can be regenerative and more long-lasting because they're based on dynamic linkages, and you can tailor the microenvironment in one step to achieve recoverable catalytic layers and enable electrosynthesis under constrained pH conditions. I realize that I've been talking very slowly, so I don't know if I will have enough time to talk to you very much about our selective electrosynthesis strategies. So in the interest of time, what I would do is simply just give you a snapshot of what we're working in that regime. So what we're doing there is we're using electrocatalyst materials to do electro-organic synthesis. And we found that you can do different types of carbon sp3 bond forming chemistries. So for example, let's see, we can take these, we can reductively and catalytically functionalize ketones and aldehydes with organic halides using electrocatalysis. And you have functional group tolerance towards different redox active functionalities, ketones, as well as sterically hindered aldehydes. And what I wanted to show you is that this is indeed a reusable catalyst. Here we're using silver as a catalyst material. If you use a silver catalyst that has seen over 30 reactions, then and you compare the yield to a catalyst that has never seen any reactions, you really see a similar isolated yield of product. So this is showing the hallmark of heterogeneous catalysis. And we can also scale this up in an undivided cell. So the workhorse catalytic strategy to make these types of disconnection or forge these types of disconnections would be Nozaki-Hiyama-Kishi conditions. When we screen those that are reported for sp3 bond forming chemistries, we don't really see synthetically tractable yields with the substrates that we've examined, suggesting that silver catalysis, electrocatalysis, could enable a complementary approach for achieving this type of reactivity. And then you can also scale this up by purposefully making what we think is an important component of this reaction by corroding magnesium in the presence of tetrabutammonium diacetate, which we believe helps enhance the efficiency of the reaction. And I can definitely explain this in further detail in the Q&A if you're interested. And so then hopefully that gives a snapshot of the type of problems we're looking at in the electroorganic space. And now I would like to conclude the talk. So we're establishing this type of research paradigm in my group where we're embracing the disorder that is inherent in the electrochemical system, designing strategies to establish more predictive control, and then leveraging them for selective electrosynthesis across a wide array of different electrochemical modalities. And in doing so, maybe we can think about ways where we go beyond the traditional catalyst development paradigm that's inherent in these molecular catalytic systems, and instead embrace the disorder that's inherent to heterogeneous systems, and think about non-covalent interactions such as molecular recognitions, van der Waals interactions, electrostatic interactions, so that we can still use electrons for sustainable fuels and chemical production without needing to use materials that aren't inherently scalable. So with that, I'd like to conclude and thank the amazing people in my group who have done all this work. The current members of the lab are highlighted in blue. Black are alumni of the lab. We're extremely grateful for all the generous funding that has supported our group. And thank you so much, especially to the Bayer Foundation for this amazing recognition. I'm deeply indebted to the energy, spirit, and passion of the people that I get to work with every day at the University of Chicago. I hope that I gave you a flavor of the type of science that we're working on. I'm happy to discuss any part of it further, and thank you again so much. And I'm happy to take any questions.